Yeah, so today is the last lecture, and I'm going to talk about modeling and simulations. And then right after, we'll have a session where we'll go through the homework. And then you can ask questions if you have any about the homework. Modeling and simulations. So often when you do experiments, you want to extract sort of the essence of that experiment. So you build models, and there are different ways of building models. What we're going to talk about is how one can build simple models, mainly and then use simulations to test those and then compare to experiments. There are a few different ways to do that. So we're going to talk about how to start with a very simple model and then add on more and more complexity to that. And we're mainly going to evaluate these models in two ways. One is Monte Carlo simulation, where you use a random number generator and the other one is we're going to look at how to solve some very simple differential equations numerically. Monte Carlo simulation. So from the name, you hear that it involves randomness. It's been applied to a lot of different things, like for example optimization problems and numerical integration, but we're not going to talk about that. We're really going to use it to sample from probability distributions. And we've already come across this a few times during the class. So you have sort of seen simple Monte Carlo simulations. So we've talked about this. So if you have an arbitrary distribution, one way to sample from this distribution randomly is to first generate a pair of random numbers within the range of the x and the y. So and then check whether this is under the distribution. If it's under the distribution, we keep it. If it's above, we throw it away. And then we continue this process and sample as many points from the distributions as we want. So this is very easy to implement, and we've used it in the past, and I think you've actually seen some of the code to do this. So this is a basic of many Monte Carlo simulations. So and what we applied it to is to try to understand some simple statistics. So for example, we looked at the central limit theorem I think it was in the second lecture, and we randomly sampled using this method from a distribution, and we saw that this is the distribution we sampled, so in this case a normal distribution, and then depending on what's the sample size. So if we sample 3 points, we see that we get a distribution that's slightly narrower, but then when we increase the sample size, we get the distribution of the mean becomes narrower and narrower, and this is scales as the square root of n, at 1 over the square root of n. So what we learn from this, we can derive this empirically. And that you can do for any time you have a question and you don't understand why something is. It might be useful to write a simple simulation to test and at least see that it works. So another thing that often is interesting in biology is diffusion. So diffusion we can simulate by a random walk. So here we start out with all the particles, in this case in the middle, and at 0. This is at time 0, and then as time increases we see that they spread. So what we do is we simply at every time step we decide whether the particle goes to the right or the left, and we then get a distribution. And we can derive the exact form of this distribution, which is again a Gaussian form where the width scales as the square root of the time. So these are now the histograms are our simulations, and the red curve is the exact form. So in this case, we didn't have to do a simulation because it was quite straightforward to derive the exact formula. But even if we add a little bit more complexity, it's not so easy to do it, to calculate the exact formula. So one seemingly very small additional difficulty would be that there is a wall at 0.3 here. So right here we have a wall that's not transparent. So if we redo this simulation, we see that particles can't go through the wall, and we get a deviation from the free diffusion. And we get quite a build-up right at the wall, and so on. And we can even add on a little bit more complexity by allowing that there is a small probability of going through the wall. So here we see that that we get the build-up at the wall here, but some of them do go through, but are not allowed to go back in this case. So this is just showing that we can, with this Monte Carlo, it's very easy to do small modifications that can be interesting if we want to build a model of a system that we're studying. So now if we look at collisions. So now what we're going to look in one dimension, and we're going to just look at let's say on DNA there are two proteins complexes that are moving at random. So in one dimension, and what's the chance that they're colliding? So if we look at a big population of cells where this happens, then we see the diffusion. So one particle starts here and one here, and then as they diffuse, we get wider and wider distributions, and we see that here we get a deviation from the expected free diffusion form because they collide and we take them out of the game when they collide. Then we can also look at what's the distance between the particles, and here in the beginning here they're all at the same distance from each other, and then the distances change, and finally we get the build-up here of the particles at zero distance that collide. So this is, and this is, as you'll see, we'll give you the code. This is only a few lines of code where you can generate all these simulations. So now, of course, we can also look at the probability. So now observe that the times here were 1 through 9, but now we're looking at a different time scale up to 1,000. So now we're looking at what's the probability of collision. So we see that, so this is in one dimension, just two particles. So what's the probability that they collide within a certain time? And it's eventually will go up to 100, but very slowly. So now there's a big difference between if we do the same simulation in 2 dimensions, then of course the probability will be much lower. So we can then add easily an extra dimension to these simulations and see that it takes much longer for the particles to find each other. So now we would have to simulate much longer times for it. Let's say if we're interested in the probability, the time it takes for half the particles to collide. So now these diffusion simulations are part of a class of models that are called agent-based models. So we have individual agents that move according to certain rules. So in this case, it was a very simple rules. They first walk at random until they collide, and when they collide, then we take them out of the game. But one can build, of course, more complex models. So one application is, for example, to look at flocking. So looking at birds or fish. So we can, with very simple rules, get behaviors that are similar to flocks of birds. So the three main rules that one would apply to each agent is that first, they shouldn't get too close to the closest neighbor, and then they should fly in the same direction as the other birds around them, and they shouldn't go too far away from the nearest neighbor. And but only using, they can only see their closest neighbors, but still we get the complex flocking behavior that we see with birds and with fish. And another application agents model has been to more understand the fundamentals of evolution. So how can we get cooperation when really in exploitation seems to be the best strategy? So this is has something that's been explored quite a lot, and it's called the prisoners, based on prisoner's dilemma. Two people being arrested and interrogated separately, and the question is, what should they do? Should they say that the other one did it, or should they just keep quiet, or say that they did it? So the conclusion is that the best, if you play the game once, the best strategy is to tell on the other person. But then if you add on other factors, like playing the game several times, playing it in a two-dimensional plane, and then you can see that eventually cooperation is a better strategy. So now differential equations. So I don't know how familiar you guys are with differential equations. So who feels very comfortable with differential equations? Okay, not very many. But you understand the basic concept that this means that a change in the change in n is proportional to n. So we're studying, like we're going to look at cell growth, for example. We call it, so these are the number of cells. So n as a function of t is the number of cells, and k is a proportionality constant that gives you the change. So what we can do is we can actually, there's an exact solution, and if there is no constraints, it will grow exponentially. But we can also solve this numerically. So there we change it not to a differential equation, but a difference equation. So here we have a small change in n divided by a small change in t is proportional to n of t. So we can rewrite this as, if we now, since we're going to do simulation, we can introduce discrete time. So we have different time points. We have n t at t of I and t of I plus 1. So that's the difference. So now we can rewrite this simply that we have the number of cells at a certain time point, and we multiply it 1 plus k times the time step. And then we get the next time point, and then we repeat this over and over again. So then if we do that, we get exactly the same. If we choose delta t small enough, we get exactly the same exponential growth. So here the red is the exact solution, and the black curve is the numerical solution. So this is what we, in general, in how we solve differential equations. And it's much more convenient in general, especially when it gets more complicated, because we just apply this simple formula like this. So now we can make this add on a little bit more complexity, because usually there is exponential growth can only go on for a while until the environment supports it. So now we're going to change this differential equation to add on another term, where we have n max is the maximum number of cells that we can have. So what you see here is if when n at time t reaches n max, this whole the expression within the brackets will be 0. So there will be no more change. So now we can rewrite this one the same way. There's an exact solution, but we don't have to worry about that. We can rewrite it, and it again becomes a simple iteration. So we start with a certain number of cells, and then calculate what the cells are at the next time point with a just slightly more complex expression this time. And now we get a curve that initially grows fast, but then saturates. And again, we get the same solution with the numerical and the exact solution. So now we can add on yet a little bit more complexity, which is now we're going to have two types of creatures. We're going to have prey and predators. So what we do that, so the change in the prey will be, so the KB is that the rate at which the prey gets born, and then KS is the search efficiency of the predators, and then we multiply by the number of the prey with the number of the predators, and that has a negative sign, because that's when the predators eat the prey. And then we have a similar expression for the predators, which they die at a certain rate, and they get born with a certain rate that is proportional to how much they can eat. So now we get, from this, now we can get an oscillatory behavior. So what happens is that the prey first initially grow when there are few predators, but then when there's a lot of prey, the number of predators go up, and they eat too much, so that the population of the prey collapses, and then it continues this way. Or we can sometimes get, so we can also show this as a phase diagram. So we here now have the number of predators on the x-axis and the number of prey on the y-axis, and this is where we're starting, and then it goes round and round because it is oscillating back and forth. Now, so this is the same figure, but sometimes, if depending on how we choose the model parameters, it can the predators can be too effective, and we can get a situation like this, where the predators eat all the prey very fast, and then so first the prey population collapses, and then with a delay the predator population, so everyone dies in the end. So here is where we start, and then we go just once around, and then stop when everything is gone. So this is then so we can change the parameters and see these different behaviors. So now we can add one more thing to this. We can add the limited resources. Same equation, but just that the number of prey can't grow exponentially, but they have a limited number of limited resources to eat. So now in this case, what we get is first an oscillation, and then both predators and prey reach one level, and it can also be shown in this diagram where we start here and then it goes and finds a point where everything is stable. So now one thing that we saw that we could do this when we chose a very small time step. But of course, if we start choosing a larger time step, we're going to introduce errors. So here is a small time step where we can repeat this circle many, many times. And it actually, the simulation here was done so that it goes around this circle many, many times. But then if we start increasing t, we see that there is a slight deviation here. It's still pretty stable, but we see that sometimes it goes further in, sometimes it goes further out. And then when we take really large time steps, we're going to get that large deviations from the harmonic behavior, and eventually, in this case, what happens is that the number of prey went down to 0 at some point. But it could also exponentially grow. So this is a very important point to choose the right time step so that one doesn't get, yeah. No, it's exponential. At some point, it's usually up to a certain level. There are many time steps that work, but at some limit, there's starting to be time steps that don't work, and then you have exponential growth. So but it's an important thing to try to find out what's a good time step, because you also don't want to make the time step too small, because then it takes forever to do the simulation. So to save time, you want it to be as large as possible, but then it can't be too large. So then we can actually apply these differential equation to understand some of the basic design principles in biology. So, for example, negative autoregulation is very common. We see that in a lot of different places, but for example, in the regulatory transcription networks. So the principle is that we have an input signal that makes a gene being transcribed. And then we have a time delay, because then this gene has to usually be made into a protein, and then it's the protein then negatively regulates the expression of the gene. So what we get is now the very simple formula where we have the input signal here. We have a constant times the input signal, and then minus, that's the negative regulation, and then we have another constant, and times the number of let's say mRNA in this case, and then there's a time delay, and that the time delay there is the time it takes to make to do the transcription, and then also to make the protein and transport it back into the nucleus and so on. So from just adding this small time delay, now depending on how we choose the parameters, we can get all sorts of so we saw before that the black curve here, so the limited resources was one way to regulate and set a level. But actually, we can use negative autoregulation to also just reach one level. And it's, in this case, it reaches it slightly faster, but we can actually, by changing the parameters so that it overshoots a little bit, we can get an even faster response. So that's one important that we can use negative autoregulation. Oh, so the input signal here is this blue line. So at an early time, we switch on the input signal and then it stays constant. So, but we then, in response to a constant signal, we can get either that we reach a regulated level, or we can also increase the response. Or for other settings, we can get oscillations. So we can, all these things we can, and this is very nice that we can, from a constant one input signal, just a step signal, get oscillations. So what you can see here is that these are, of course, very desirable in biological systems. That you can be able to do all these different kinds of transformations of signals. Then we actually have one other very common network element. It's called a feedforward loop. So what it means is that X regulates Y, which regulates Z, but then X also regulates Z. So as you can see, there are three different connections, and so each of them can be either that it's a positive or a negative regulation. So we have 8 different possible ones, and 4 coherent and 4 incoherent. But what turns out that in reality, in bacteria and yeast at least, what's mostly used is one of the coherent ones, where this one and one of the incoherent ones. And these can act also as additional transformations of signal. So the coherent one can filter out spikes, which is of course important, so that if there isn't in a noisy environment, we want to be able to not respond to short spikes, but longer signals we want to be able to respond to. Then we can also set the parameter so that it prolongs the response. And the incoherent one can act as a pulse generator. So if we give it a step function, it will give us a pulse out. And there are actually more properties that we can look at. So these were sort of now I'm just going to go into tell you about a few examples where this has been used. And the first one is an HIV progression and transmission model that people at the population health here have developed. So there are 2 models. The first one, the progression model, is an agent-based model where we simulate lots of individuals and where they can change the way that the different, so they're depending on what their viral loads and CD4 counts, for example, are. They can go from they have a certain chance of dying, or they have a certain chance of changing their viral loads and so on. And also, of course, the treatment has a big difference. And then the transmission model is a flow model which has different compartments. And the main thing is that you're either susceptible, infected, or immune in this case. And then, so if we look a little bit more in detail, so what we do is we, in this, we have data from by collecting information about how people progress. And so that it's usually quite difficult to collect a lot of good information about how people progress, but we can. But the nice thing is then with the model is that we input all these probabilities, how it is to well, if you have a certain viral load, what's the chance of dying if depending on your age. So we get, but then we can simulate a much larger population, and we can try different interventions in the model. So often what they use a model like this for is to ask questions like, if New York City has a few $1 million that they want to spend on HIV intervention, what's the best way to use that money? And so this is again, it's a Monte Carlo simulation where we at the next event that can happen to an individual is determined depending on what kind of state that individual is in. And so you can do the simulation, of course, in a resource-rich environment or in a resource-poor environment and see how that. And then this progression model feeds into their transmission model, where they have in the transmission model, people are born with a certain probability and then a certain rate, and then as soon as they're born, they're susceptible to being infected. And then there are these rate constants to move from different, but there are not only 3 different categories. There are different boxes for different ages, gender, sexual orientation, sexual activity, CD4 count. So it's an enormous amount of different sort of boxes that people can be in, and then you calculate the flow between these. So another type of simulations that people do is called molecular dynamic simulations. So in this case, we again solve a differential equation, which is Newton's second law of motion, which says that the mass times the acceleration is equal to the force. So here we can again make this into difference equation by realizing that the second derivative can be approximated by a symmetrical function. So what we get is that the position at time tn plus 1 can be calculated from the 2 time steps before and the force on the particle. And so the question then, how do we calculate? So usually molecular dynamics are used for a lot of cases. For the most interesting for us is probably related to protein structure. So we determine protein structure by crystallography in NMR, but often we want to then look at how 2 proteins whose structure we know interact. What's their interaction surface? So one thing we can do is we can then calculate. So we need to define what's the force on each particle. So here, if we have the energy landscape, and that we can do if we know the bonds, if we know, and we have to make certain assumptions. And so, for example, for a covalent bond or an angle between 2 bonds, we can have a harmonic function that can be done. Then we of course have the electrostatic function, which goes as 1 over r, which is a very long range. And then we have the van der Waals interactions, which is an extremely hard atom and a small energy minimum. And then we can also simulate the covalent bond and the angle with a more realistic potential where it's actually possible to break the bond. So this is just one example from Stephen Hubbard's work. where they looked at the interaction between two domains of the JAK2 tyrosine kinase. So what they did was they simulated the box with a side of 120 angstroms and using periodic boundary conditions. So if one atom went out the other way, it went in from the other side. And they had 165,000 atoms, and they also had lots of water molecules and some sodium chloride ions. So again, they used this electrostatic potential and this other Lennard-Jones potential for the bonds. So then, in this case, they tried to different configurations and moving these together and simulating all these atoms for about 3 microseconds. They tried it 14 different times and then found a nice structure where that looked like a real interaction surface. And then they continued to do a lot of other calculations. So this now, these last two examples, both the HIV progression and transmission model and these molecular dynamic simulations, require a lot of computational resources. But that's not true for all models. So here we developed a model to look at how to design proteomics experiments. And mainly the idea is that in proteomics, you usually observe the high abundant proteins, and it's very difficult to get down in to the low abundant. So this just shows what, as a log protein amount, we have roughly a distribution. There are few high abundant proteins, a lot of in the medium abundant range, and fewer in the low abundant range. So what we did was to simulate this workflow. And there are lots of parameters that one can look at, but the main thing is that we don't really know a lot of these parameters in the simulation. And that's true for many of the models that we're making, that a lot of the input we don't know. So the main trick there is then to, if there's a parameter where you don't know what its exact value is, the best is to try a wide range. So you try very low and very high, and preferably even at whatever you think is the highest possible or lowest possible. And that way you know if you observe something that's stable over that range, then you can conclude that your model, then you can say that your model is useful. But so this just shows one example of when we do repeat analysis. So if we do one measurement on a proteome, and then we continue to do more and more of the same measurement. So what we see is that we see more and more of the high abundant proteins. So the yield, the success rate here of our measurement increases. So this is just comparing simulation and experiment. But actually we don't go further down into the proteome at all. So I'm going to skip this and move to the next one. So we talked a little bit about that we also used molecular dynamic simulations to analyze images. So and especially we looked at overlap. So in this case, we did Monte Carlo simulation. So we take the properties that we measure of the particles, so size and density and shape, and then randomly put them in a box and look at the overlap. So this shows a little bit more detail. So these are the distributions. So we have two proteins, connexin 43 and plakophilin 2. So this, for connexin, we see here, this is the distribution of the areas, of the ratio of the major and minor axis, and then we using the method that we draw from this distribution, and this just shows that we actually succeed to sample the distributions well, and then the same for the other particle. And so then, if we then randomly put these and then look at now this slice where this is connection 43 area and this is co-localization area, and we just look at the particles within this range and separately the particles here, we see that if we the experiment measurement is up here and our simulation is this point, so we're far away. So the experiment, what we concluded that what we see is it's not random in this case. So but the next step then, of course, is to add on that we can add an attraction between these, that there is a higher probability that they actually co-localize. And then we can see that here, if we increase this attraction quite a bit, we can get an overlap with the experiment. And then when this other protein, anchorin G, is silenced, we need a much smaller attraction. So what happens if anchorin G is silenced? We're moving towards more randomness, but it's still not random. So a final example now is where we looked at replication fork progression. So we looked at yeast and we took one of the subunits of the GINS complex, which is associated with the replication fork, and tagged it, and then we took time points throughout the cell cycle and measured, did the chip-chip experiment. So this is just looking at chromosome 16. So when we are in G1, nothing much happens. We see mainly noise. Then when we enter S phase, we start seeing binding of genes to the to the origins of replication. And then first it binds, then it accumulates and starts spreading. And then here now it has merged with this the neighboring one. And then at the origin, there is a dip finally because it's done and it has moved out. And then it continues, and finally it will again not bind to the DNA. So we see this is just chromosome 16, but we see the same thing for all the chromosomes. So in summary, we can see 303 active origins fire in the first 10 to 15 minutes of S phase. And then once they fire, they move at a constant rate. And the moment, and it looks like it's symmetrical. So now what we can do is make a simple model out of this, just implementing this. So we have the binding at the origins, and then they start moving out, and when they collide, they're going to fall off because they're done. So this is an extremely simple model, and we can now quite easy to implement this as an agent-based model. So they have we have the binding at different origins, we have a velocity, and we have a certain timing. So what we can see now, if we compare experiment to our simulations, that we see a very similar behavior. So this is a region with 3 origins, and one starting a little bit earlier than the others. And then we see the progression, both in the experiment and the simulations. Another more complex region with 7 origins. We see at telomeres, it doesn't seem to be any different. At centromeres, also that doesn't make a difference. And these are the 2 origins that are furthest apart in yeast. So in conclusion, we can see that, I mean, it wasn't extremely simple model, but we could still understand our experiments much better after doing this model. So in summary, I talked a little bit about Monte Carlo simulations and how one can use these to make simple models. We talked a little bit about differential equations, about both how negative autoregulation feed-forward loops are important biological building blocks. And then we looked at a few different examples. So this is sort of the end of the course, and now we'll talk a little bit about the homework, and then on Thursday is the exam.